This unit will cover skeletal muscle, tendons, and ligaments. These are the session objectives that you should address when studying this material. We've looked at muscle tissue before when we studied cardiac and smooth muscle a couple of blocks ago. Today we're going to look at specifically skeletal muscle, but let's start at the beginning. Remember, muscle is a tissue that's specialized to contract through the shortening of individual muscle cells. This contraction can be a source of heat. Also recall that there are three histologically distinct muscle tissue types or subtypes: skeletal, cardiac, and smooth. What are the characteristics of skeletal muscle? It is specialized for contractility and excitability, and these are in order to perform voluntary movements, that is, in contrast to cardiac and smooth, which are both involuntary. It's a type of striated muscle, and it shares that characteristic with cardiac muscle. And its organelles and cytoskeletal arrays are very precisely organized in order to contract. Skeletal muscle is under the control of the somatic nervous system and has both components of the sensory and motor systems. As we learned previously and will review in a minute, muscle tissue structures have unique names. So what is that specific terminology? Well, muscle cells can be referred to as myofibers and muscle fibers. Remember, the myofibril is an organelle specific to striated muscle with repeating sarcomere units. These contractile sarcomere units are comprised of myofilaments, which are the cytoskeletal filaments, including actin, myosin, and all of their accessory proteins. The plasma membrane of a myofiber is often referred to as the sarcolemma, and the cytoplasm as sarcoplasm. The smooth ER in muscle fibers is often called the sarcoplasmic reticulum. Recall that this sarcoplasmic reticulum ensheathes the myofibril organelles. How is skeletal muscle organized? Let's start at the largest gross muscle level and work our way down. So large named muscles are divided into groups of muscle fibers called fascicles, and those muscle fascicles are comprised of individual muscle fibers which contain multiple myofibrils of repeating sarcomere units that are made up of those contractile proteins. How does this look histologically? So the muscle longitudinally can be seen to have numerous fascicles, which are multiple muscle fibers bundled together in a connective tissue covering. We're going to discuss those coverings in more detail in a few minutes. At a little bit higher magnification, you can see a muscle fiber or myofiber, which is a multinucleated cell. These cells have peripherally located nuclei as well as cross striations due to the regular arrangement of those myofilaments inside of them. Those two features of peripherally located nuclei and striations make this particular tissue a little bit more easily identified. How does that kind of a structure come about? So initially, the precursor cells called myoblasts proliferate and align themselves and join up by gap junctions and various other junctions. They eventually fuse to form a larger structure, which is multinucleated, and it's called a myotube. At this point, the nuclei are still centrally located. They are not yet peripheral. This structure will further differentiate into a more mature muscle fiber, when the contractile apparatus develops and the nuclei get pushed to the very periphery right underneath the sarcolemma, you end up with very long multinucleated syncytial fiber-like cells. How does this look? On the left you can see a cross-section of myotubes, and on the right a longitudinal section. And I think the longitudinal section, you can appreciate that these cells still have centrally located nuclei because the contractile apparatus is not yet developed. It has not yet matured into mature skeletal muscle fiber. Now let's take a quick look at those connective tissue coverings. So on the very outside of an anatomical muscle is a dense irregular connective tissue layer called the epimysium that surrounds the entire muscle and bundles all of the fascicles together. Inside of that, the muscle fascicles are bundled into their little groups by another thinner layer of that dense irregular connective tissue called the perimysium. Within the fascicles, individual muscle fibers are ensheathed by a very delicate fine layer of reticular fibers, and there's fibroblasts in there too, called the endomysium. So these connective tissue coverings are where you're going to have your vascular and nerve supply to that muscle. How does all this look? You can see at the far left the outer covering of the entire muscle, the epimysium, and you can see numerous fascicles at low magnification and some evidence of the perimysium. I think that the fascicles are very easy to identify in cross-section on the right here, and you can see how they're grouped together, and fortunately there is some artifactual separation between the fascicles, so you can distinguish them a little bit more easily than maybe you could do in the longitudinal section. At higher magnification, you can see muscle fibers with multiple peripherally located nuclei. And these are apparent both in longitudinal and cross-section, and are one of the really giveaways that's going to help you identify this tissue as skeletal muscle. You can also see the vasculature in the perimysium between fascicles, and the details of that vasculature, such as endothelial cell nuclei, are even distinguishable. At very high magnification, you may even see a little bit of that endomysium. You can certainly see some fibroblast nuclei present, as well as the evidence of the vasculature, such as capillaries. In longitudinal section, the cross striations really jump out at you and are again one of those diagnostic features for which tissue you're looking at. Let's talk for a minute about muscle fiber types. There are 3 different histochemical types of skeletal muscle fibers, and that's based on their differences in the contractile speed and duration of the contraction, as well as fiber morphology and the metabolic profiles. Most muscles have all 3 fiber types within them, but the proportion of which one predominates depends on the function of the particular muscle. That will be discussed in detail in the muscle physiology didactic material. Let's just briefly take a quick look at them without going into great detail. First, you have the slow twitch muscle fiber type, also called type 1. These are specialized for small amounts of force over prolonged periods of time. Therefore, they are also called fatigue-resistant fibers. Now, on the other side of that spectrum are the fast-twitch fatigable, or type 2B fibers. These produce large amounts of force for very short time durations. In between those two, you have fast-twitch fatigue-resistant, which are type 2A fibers, and they produce moderate amounts of force for moderate durations. So the details of how these work and what the differences in their metabolic profiles are will be covered in more detail, as I said, by the physiologists. Let's talk about the myofilaments for a few minutes. These include both the thin filaments of actin and the thick filaments of myosin, and all of the associated proteins for each of those. Now, thin filaments and their associated proteins are really three different proteins. Actin, of course, which is a globular protein which polymerizes into a long filament, F-actin, and in muscle is a double helical structure. Tropomyosin, which is a protein also organized into a double helical structure, and that lays along the groove of the actin helix and acts with another molecule, troponin complex, to regulate the actin-myosin binding. And the third that I just mentioned is that troponin complex that has three different globular subunits. TNI, which is an actin binding protein, which actually prevents the binding of actin to myosin, that's regulatory. TNT, a tropomyosin binding protein, which anchors that troponin complex to the actin, and TNC, which is a calcium binding protein, which is very important in the initiation of muscle contraction. This is how those are organized together. So actin is the light blue there, and the long rod-like tropomyosin is lying in the grooves, and the troponin complex is organized in various intervals along that filamentous strand. The thick filaments, or myosin, also has three different major proteins. This is comprised of myosin heavy chain and myosin light chain 1 and 2. The myosin heavy chain is very large, has a long rod-like tail section and two head sections, which are the ones that are ATP binding and actin binding. So that's where the movement and attachment occurs. The phosphorylation of the light chains regulates calcium stimulation of contraction. You can also see that all of those long rod-like regions of the heavy chains are all associated together to form that very large thick filament, and the head regions are kind of sticking out all along the length, which facilitates the interaction with actin. Now, myofilaments within the myofibrils are linked together in this very organized fashion to form the sarcomeres. And the sarcomeres are organized end to end to end, all the way along the length of these fibers to form myofibrils. So what is a sarcomere? Recall from our prior study of striated muscle that a sarcomere is the unit of contraction. At the light microscopic level, you can see the A band, the dark, and the I band, the light regions, which alternate AI. Bisecting the I band is something called the Z line or Z discs, which is the place where all of the thin filaments are anchored. So a sarcomere goes from Z line to Z line, or Z disc to Z disc. At the center of that A band, there's typically a lighter area called the H band, where you do not have overlap of myosin and actin. So it's just a myosin area, and that is bisected by something called the M line, where things are organized. The myosin thick filaments are kept in register and organized with accessory proteins.